Bismillahirrahmanirrahim. 9700 AS level biology paper 2. And now this is the second video for the June 22 paper 2-1. Sorry, not 4-1. For 2-1, and this is question 4, 5 and 6. And now looking at question number 4, adipocytes are cells found in adipose tissue in mammals. These cells absorb glycerol and fatty acids to make triglycerides for long-term storage. Figure 4.1a shows a glycerol molecule and 3 fatty acids. 4.1b shows the triglyceride molecule formed from these components. Now you have figure 4.1a and figure 4.1b. State the name of the bonds that form between glycerol and fatty acids, ester bonds. So this is actually a part of the syllabus in which they ask you for this. So ester bonds. Then when a bond forms between glycerol and a fatty acid, water is a product of the reaction. State the name given to this type of reaction. When water is given out, what is it called? Condensation. When water is needed, the opposite of this is hydrolysis. Everybody must be very clear on hydrolysis and condensation. So when water is a product of this reaction, when water comes out of the reaction, when a bond is formed, so that is called condensation. Describe the differences between the fatty acids X, Y, and Z. So you're only asking for this X, Y, and Z. The differences, it says differences between the fatty acids X, Y, and Z. Now, if you look at them, what are the differences? Very simple. Different lengths. Y is longest and Z is the shortest. Y is the longest and Z is the shortest. You can see here, Y is the longest and Z is the shortest. Then Z is saturated. What we have to understand is Z is saturated. There's no double bonds between carbon-carbon. So carbon-carbon has a double bond. So Z is saturated, X and Y are unsaturated. You can see this is the double bond which you can see here, and then here, and then here, and then here. So 3 double bonds in X and 1 double bond in Y and no double bond in Z. So X is polyunsaturated and Y is monounsaturated. X is polyunsaturated. Why? Because of 3 double bonds. And Y is monounsaturated. So that got you another mark. Or you could have said X3, Y1, Z0, double CC bonds. Or you could have said X is 4 double bonds, Y is 2 double bonds, and Z is only 1 double bond for whole molecule if you had to talk about. So how will you write this? Y is longest, or you can say Z is the shortest, and Z is saturated, X and Y unsaturated. X is polyunsaturated, Y is monounsaturated, and X is 3, Y is 1, and Z is 0 double CC bonds. Then coming to the B part, one of the questions state reasons why triglycerides are described as hydrophobic. So basically you've got to describe what is hydrophobic. The fatty acid tails are non-polar. They have no polar groups. They're not charged. That is why they will not be, they're not soluble in water. And they cannot form hydrogen bonds with water. Basically, why is sodium chloride soluble is because it forms hydrogen bonds. So this is what you have to understand is why they are called hydrophobic. Fatty acid tails non-polar, not charged, have no polar groups. You can use any of those words. So they are not soluble in water and they cannot form hydrogen bonds with water. So any 2 out of these 3 would get you your 2 out of 2. Then coming to part 2, explain why triglycerides are not suitable as a component of cell surface membrane. Now, in order to answer this question, you've got to understand how is the cell membrane made up. It's a phospholipid bilayer, but there's no phosphate here. There's no phosphate head. So there's no hydrophilic, polar, or phosphate head here to interact with the water on the outside. Every cell has fluid outside it, water outside it, watery medium outside it. In humans, it's called the tissue fluid. And this cannot form hydrogen bonds with water, and it cannot form a bilayer. So that is the reasons why this cannot be a phospholipid bilayer. So basically, no phosphate had to interact with the water, cannot form hydrogen bonds with water, and cannot form bilayer. So, I mean, even if you didn't know it, you could have thought of the cell membrane. What are the structure and the functions of the cell membrane? Now, these cannot form the cell membrane because the cell membrane is a bilayer. Plus, it has to have a hydrophilic phosphate head, which interacts with the water outside. Then coming to question number 5a, infectious diseases are caused by pathogens and described as transmissible diseases. Explain what is meant by the term transmissible. That means the pathogen can be transferred from an infected to an uninfected person. Transmissible means you can't use the words of the question. You can't say transmission. You could say transferred. So basically, the pathogen, which is the causative organism, can be transferred from an infected person to an uninfected person. That is the definition of transmissible. Then it says Vibrio cholera is the bacterium that causes cholera. Figure 5.1 is a transmission electron micrograph. The flagellum bacteria. Student who wanted to know the actual length of the flagellum shown in figure 5.1, state the information that is needed so that student can calculate the actual length of the flagellum. What is needed? You need the magnification of the image of the electron micrograph. So you need either a scale bar, and then you need to calculate the length of the flagellum, and then you use the formula. What is the formula? It's very simple. The formula is actual is equal to image over magnification. So you just use the formula to calculate the length, measure the length of the flagellum and divide it by the magnification, and that will give you the actual length of the flagellum. So transmissible pathogen transferred to an uninfected person, and here we needed the magnification of the image, then we needed the length of the flagellum in the image on figure 5.1, and then we use the formula actual is equal to image over magnification. So you could have written the formula, you could have written it in words, or you could have written it even like this, and they would accept it. Even if you did the magnification triangle, even that was okay. Part C of the question table 5.1 shows data on large outbreaks of cholera that occurred in 7 countries in 2019. The case fatality rate is the number of deaths as a percentage of the number of cases. So number of deaths as a percentage of the number of cases, and this is called the case fatality rate. Now it says complete table 5.1 by calculating the case fatality rate for Yemen, which was not done. So you have to divide 3767 by 2260495 and then into 100, and you got this, and it was only two. You can see 4.2, 1.7, so you had to only one decimal figure, so you have to only give me that. Now comes, of course, a very long and lengthy question. Suggest why large outbreaks of cholera occurred in the countries listed in table 5.1, but there were very few cases in most other countries. That is 5 marks. So you've got to know the whole story of cholera and why would there be many more cases of cholera. Because basically you've got to understand cholera is a waterborne and a foodborne disease. So civil disturbance, civil war, or you could say of natural disaster, lack of uncontaminated drinking water, and sewage disposal is not very good. Then there's poor sanitation, then the personal hygiene is not very good, then eating contaminated foods, or you can say crops are irrigated with contaminated water. Then there's a lot of time for diagnosis. People do not have access to health care, and then they do not have oral rehydration salts are not provided, or they are not available free. People don't have the money to buy them. Then antibiotics are not provided, or they're not available. Then the vaccine is not provided. Then poor understanding, education, prevention of the disease, and it's difficult to reach the people living in the remote areas or the rural areas, and there's lack of professional medical people, and there's people suffering from antibiotic resistance, or people coming in from some other place from nearby and bringing in the disease from other cities, coming to those places, or coming from other countries to those places. So about 14 mark scheme points, and any five of those would get you your 5 out of 5. So now looking at the verdict that you would use for this is civil disturbance or war, which causes, the infrastructure to be destroyed. Then, if you said there was a natural disaster like an earthquake, or some sort of floods or something, then lack of drinking water, then sewage treatment is not available, no separation of drainage from homes and water supplies, and contamination of water supplies with human feces, then poor sanitation and poor personal hygiene, eating contaminated food like crops irrigated with contaminated water, seafood harvested from contaminated seawater, and then there is slower or no or little diagnosis, then there is no or little treatment of healthcare and medical facilities, then there is oral rehydration, salts are not provided or not available, antibiotics are not available, vaccine is not provided or the vaccine is not available, then poor understanding and education about the prevention and treatment, and difficult to reach the people in the rural areas, lack of trained professionals, antibiotic resistance, and people bringing in cholera from elsewhere. Question 6. Co-transporter proteins are membrane proteins found in companion cells of phloem tissue. Messenger RNA is the molecule in cells that carries genetic information in the DNA that codes for co-transporter proteins to the sites of protein synthesis in the cytoplasm. Complete table 6.1 to compare the structure of a molecule of mRNA with the structure of a molecule of DNA. Name of the four bases. Now, of course, name doesn't mean that you just write the letters and you just say A, U, G, C, and here you write A, T, G, C. Well, no, you'll get no marks for such a thing because name means you give me the names, you don't give me abbreviations. So this would be all rubbish and you get a big fat zero for that. So you give me the name, so adenine, uracil, cytosine and guanine, adenine, thymine, cytosine and guanine. So add GC and this is AUGC. The name of the pentose sugar in RNA it is ribose and in DNA it is deoxyribose. The number of strands is 1 and in DNA it's 2, and it said if you wrote single, double, well of course we gave you 0 marks for that because not single, it's one number of strands. Number means something else. 1, 2, 3, 4 is the number. What is your phone number? You give me a number, you don't say single number or 3 digits of 7 digits. So this is how you would have done this, and you would get for each line you would have got a mark for that. So that would get you your 3 marks. Then it says B part, figure 6.1 shows the events that occur in the nucleus of a companion cell in phloem tissue to synthesize molecules of mRNA. So we have the DNA here, then we have stage 1 and something else is produced which is actually called pre-mRNA, and then the mRNA is produced and this is stage 2. So stage 1 and stage 2. Now name stage 1 shown in figure 6.1. That was easy. Transcription. I always say it's the script of a place, so script of a protein molecule. So transcription was the answer to that. Describe what happens at stage 2, shown in figure 6.1 to shorten the length of the RNA. What has happened? Well, what has happened? The introns have been removed from the primary transcript, and then the exons have been joined together. Or you could have said splicing of the primary transcript. And I was just checking a test which I had given this question, and I had some weird answers from a large number of students whom I think didn't really know what they were talking about. So what do we have? Removal of introns and then the exons joined together. Removal of introns and exons joined together. Please remember, introns are the ones which are the non-coding regions. And you get this all mixed up. You, instead of introns, you write exon. Well, that's all wrong. I can't give you marks for that. Part C of the question says co-transporter molecules are proteins produced in the companion cells. Figure 6.2 shows what happens in the cytoplasm of a companion cell to a transfer RNA molecule before the co-transporter proteins can be produced. So we have here this structure, and then something happens, and then we have this structure. Describe the role of the transfer RNA shown in figure 6.2 in the synthesis of a co-transporter protein. Whether it is a co-transporter or any other protein, whether it's an enzyme, whether it's hemoglobin, whether it's myoglobin, basically it's the same thing, transcription and translation. So if you know that, I'm sure you can give it, but if you don't know it, then you're going to give me some weird stories. So describe the role of the transfer RNA. That's 4 marks. What is the role of the transfer RNA? The role of the transfer RNA is very clear. Amino acid activation. What is amino acid activation? It is the amino acid changing the amino acid before attachment to the tRNA. This is called amino acid activation. Then the tRNA attached to a specific amino acid. Then there's anticodon identifies the specific amino acid. Then the tRNA transport the amino acid to the ribosome. Then the tRNA entering the A site in the ribosome. Then the tRNA has the anticodon which binds to the codon on the mRNA. So if it is CCG, it binds to the mRNA GGC. Then specific amino acid in the correct position in the polypeptide chain. Then the tRNA is recycled or reused. And then if you talk about the amino acid tRNA synthetase, which is responsible for the activation of amino acids. So, I mean, these wordings have to be learned by you all, and you need to memorize these. Some of these, some of you are very good, but some of you need to really, build up this vocabulary for the biological English. So, the first point is amino acid activation. Then tRNA attached to specific amino acid. Then anticodon identifies the specific amino acid. Then the tRNA transports amino acid to the ribosome. Then the tRNA entering the A site in the ribosome. There are 3 sites in the ribosome. And this enters the A site in the ribosome. The tRNA anticodon binds to codon on the mRNA, and then the amino acid is in the correct position in the polypeptide chain, and then the tRNA is reused. Now, just a quick revision. What is the structure of a ribosome? It's got a large ribosomal subunit and a small ribosomal subunit. And then it's got the E site and the P site and the A site. A is the amino site tRNA, and then P is the peptidal tRNA, and E is the exit site, where it's going to exit. The tRNA is going to leave it. So it's going to enter at the A site. Then what is the process of protein synthesis? DNA in the nucleus as a template. mRNA is processed and released into the cytoplasm. mRNA binds to ribosome. tRNA carries the amino acid to the mRNA. Anticodon, codon, complementary base pairing occurs. Peptide chain is transferred from the resident tRNA to the incoming tRNA. tRNA departs. Protein modification after translation. Then the ribosome sites, largely based on the rRNA structure, large, small subunit, and the APE sites. Then steps in protein synthesis, DNA molecule unzips where the desired gene is located. Free-floating RNA nucleotides pair with the DNA strand forming mRNA. The mRNA leaves the nucleus, goes to a ribosome. A specific tRNA delivers specific amino acid to the ribosome. This is translation. The mRNA codon matches with the tRNA anticodon, bringing the amino acid in its proper place. When the next amino acid is in place, the two are joined in a condensation reaction. The process is repeated until a stop code is read and a complete protein is formed. Now, coming to the last part of the question, outline the role of co-transporter proteins in companion cells. What do they do? The movement of sucrose with the H ions into the companion cells, and then the sucrose moved from the cell wall or the mesophyll cell. Then the sucrose moves against the concentration gradient into the companion cell. Then the sucrose is polar, so cannot pass through the membrane, and then maintains the concentration gradient for sucrose between the companion cell and the sieve tube element. So basically, you have to give me the story about how the sucrose will move in with the hydrogen ions. So the hydrogen ions are pumped out, and then the hydrogen ions move back in and will bring sucrose along with them. And the sucrose is coming from where? From the cell wall. And the sucrose moves in against its concentration gradient. And this needs a very important co-transporter protein. And the reason why it cannot move in is because sucrose is polar, so cannot pass through the hydrophobic part of the cell membrane. And this maintains the concentration gradient for sucrose between the companion cell and the sieve tube. And once it enters the sieve tube, then the water follows, high pressure, and then somewhere in the sink, then there is the sucrose is unloaded, water follows, low pressure, and so for this movement from high pressure to low pressure is called mass flow. But of course, that is not being asked in the question. I just revised that with you. So just the wordings of it, movement of sucrose with the H positive ions into the companion cells, and the sucrose moves from the cell wall, or you could have said from the apoplast pathway or from the mesophyll cell, and then the sucrose moves against its concentration gradient. And the sucrose is polar, so cannot pass through the cell membrane, maintains the concentration gradient for sucrose between the companion cell and the sieve tube element. So any three out of these, and you would get a 3 out of 3. So this completes this paper, and I wish you all the best. And thank you once again for watching, and thank you for subscribing. And please do leave your comments as to what else do you all want me to do for your upcoming exams. Thank you very much.